DNA binding proteins meet their mismatch is the paper that I'll be looking at. Um, so first let's talk about what is the difference between a mutation and a mismatch. A mismatch basically refers to a single nucleotide alteration in one strand of the DNA. However, a mutation is actually a single nucleotide base pair um, change that actually happens in a double DNA helix. So, in this case, as you can see if you're following my cursor, here AT changes to CG, which is actually a mutation, and it maintains the alignment in the DNA, which means that it retains the complementary base pairing and it obeys the Chargaff's rule over here. However, in the case of the mismatch, only one single nucleotide on one specific DNA strand changes, which means that over here, T changes to C. And you can see some examples of DNA mismatches over here. So there's T with G, C with A, T with G, and C with A here. So in this case, this is an example of C with A. And this actually results in the loss of the alignment in the DNA double helix. And due to this mismatch, there is a distortion in the DNA. Now let's look at how this distortion actually affects proteins. So there are certain kinds of proteins in biology known as DNA binding proteins, and they are extremely abundant in biology. These proteins have been found to have greater affinity for DNA that is distorted or that is engineered to have a single nucleotide mismatch. And by distorted, I mean DNA that does have a mismatch. And when there is a mismatch in the DNA, that results in DNA distortion. So, since A is not complementary with C, it results in distortion. It results in DNA distortion, and that makes it easier for proteins to kind of fit in with the DNA molecules. Basically, DNA distortion can be seen as an analogy for a hypothesis like the induced fit hypothesis, where an enzyme is induced to allow its substrate to fit in perfectly with it. So here, the distortion of the DNA leads to the protein fitting in with the DNA absolutely perfectly. It's like a jigsaw puzzle. So the protein and the DNA fit more perfectly after the DNA is distorted rather than the DNA not being distorted. And some examples of DNA binding proteins could include transcription factors that actually regulate gene expression. And this makes their work so much easier because now they no longer have to induce a change in the DNA, and they no longer have to do any work or pay any energy cost to allow the DNA to distort or change shape to allow the protein to fit. All that work is done by the mismatch itself, and it just takes one single nucleotide to change for that to happen. So, this does indeed prove to be a very helpful technique to allow proteins to bind to DNA. Let's look at the mechanism through which this happens, and the ability of proteins to indirectly recognize 3D shapes of DNA and to bind to them is actually known as shape readout. And by shape readout, we actually mean that it recognizes the overall shape. So, for example, if the DNA has a major or a minor groove, that could be recognized by a specific protein, and the DNA and the protein could actually go and attach to that part of the DNA. But there is another type of readout that most proteins follow, and that is the base readout. And this is a direct recognition. And here, there are certain sequences in the DNA that are recognized by the protein and that allow it to bind to that part of the protein. But in case of DNA distortion that results from mismatches, the proteins follow the mechanism of shape readout because there is an overall change in shape, and that's why they cannot specifically recognize the base sequence anymore because that is no longer registered by the protein. But what is the shape. And that's what helps proteins to bind to their complementary DNA molecules. And a mismatch also improves the binding specificity of a protein because it does all the work, as I spoke before, about paying the energy cost that is no longer to be paid by the protein. All of that work is done by the mismatch itself. Now, coming to the saturation mismatch binding assay, which is a technique that was developed to avoid the assumption that all DNA is correctly base paired. So, most techniques that are used in biology assume that DNA is correctly base paired. But in this case, DNA is not correctly base paired, and it does not obey the Chargaff's rule because you do not have 100% complementary base pairing in your DNA because there might be a single nucleotide mismatch, and that's what we have to study. So, for that, they created this new technique which is known as saturation mismatch binding assay. And in this, we use a microchip array, which is as shown here, if you're following my cursor, is used where single DNA strands of every possible single nucleotide variant of a consensus sequence is placed at a known coordinate. So basically, we have individual DNA strands that might have single nucleotide variants of a consensus sequence, and by that I mean a mismatch. And for example, if I placed one of them over here, and then one I placed over here, and then one here, and then at random coordinates, and then I sent in complementary DNA probes to hybridize with these DNA strands. After hybridization, we introduce fluorescently labeled proteins that go and bind to their DNA molecules that they are complementary to. And that will only be possible if the protein and DNA affinity is enough for the protein and DNA to bind. The results of this experiment show that a DNA mismatch could indeed improve protein DNA binding by many fold. And this can also be seen if we double the number of DNA strands that are introduced with a single nucleotide variant for the first time, and in the second time, we double it. So that will tell us if there are double the number of Lydate sections, that would mean that protein DNA binding is actually directly proportional to the single nucleotide variants consensus. So, according to African and his colleagues' experiments, 10% of DNA protein affinity was improved by mismatch. And the mismatch may be in the natural target sequence or a different sequence, which means that at times DNA tends to bind to a specific protein, and if there is a mismatch, then the protein may bind to a different sequence because that then becomes the more fit sequence for the protein to bind, and that improves the affinity of the protein and the DNA for each other. And as I spoke before, that the energetic cost of distorting the DNA for protein binding is paid for the mismatch, preventing the protein from doing it, which means that the protein no longer has to do any work to bind to its complementary DNA molecule. So, indeed, this technique does come with a lot of advantages, but at the same time, there are a few associated drawbacks that is there with everything. So, mismatches may lead to deadly diseases such as cancer, and they might promote the binding of a transcription factor at the wrong site. This is because single mismatches do promote binding of proteins, and this may be at the wrong site, resulting in misregulation of genes or even the initiation of the production of proteins that allow excessive cell proliferation, resulting in the formation of irregular masses of cells that may be malignant tumors. And this remains even if the mismatch is fixed. So even if we fix this mismatch, the malignant tumors won't go away, and the cancer will not be able to be controlled. In certain cases, this also may result in the inactivation or the underproduction of certain proteins because the transcription factors may be binding at the wrong sites and they may not be able to initiate a protein production as they may overlap certain genes, and that could result in certain other deadly diseases or maybe even deficiencies in the body. So we have to keep that in mind. And other than that, overall, this technique is very helpful in terms of allowing proteins to bind with their specific DNA molecules without having to do much work, because all of the work is done by the mismatch itself. And it is certainly an advancement in science, and it's extremely helpful in terms of helping DNA molecules bind to their proteins. And that would be it. Thank you.